this afternoon, we're going to be talking about Galaxy and then actually using Galaxy with microbiome data. But to start, let's talk about what even is Galaxy. You heard about it briefly from Carrie when she was discussing cloud computing. And Galaxy is a cloud platform that has two different interfaces by which you can interact with it. One is a web UI, or sort of a GUI, a point and click. So it's a website user interface that lets you point and click, see exactly what you're doing. You don't have to be typing commands and telling it like specific syntax for that. The second is a programmatic API, so that typing commands. It also has data sets that are publicly available that you can access from Galaxy, or it allows for you to upload your own data sets. It currently supports thousands of analysis tools and visualization techniques. If a tool isn't available, you can wrap it yourself or ask the support forum to bring the tool in for you. But not only does it have all that access to data sets, it has access to a lot of tools already. And the platform is built to use computing resources that are stable and has a lot of computing resources available for you. So as Carrie was suggesting, if your computer just isn't enough, if you just don't have enough resources, as Greg was saying, you're going to move into analyses that require really big files and or going to need a lot of computing power, Galaxy can be an option for you because it has that computing power available and it can run it without overtaxing your laptop. Galaxy looks like this, taking this idea of tools, data, and bringing it all together. When you open Galaxy, it has three panes. On the left in orange, I've highlighted the tools pane. And so this tools pane lets you search tools, and you can either use the search bar at the very top, or you can use what you know about your tool. You know that it's a bioinformatic RNA-seq tool. So you might look for RNA-seq. You might know that you need to manipulate a FASTQ file, so you can look at that FASTQ thing. Either way, that tool panel allows you to search and find the tools, and you would click on it there. Once you've selected a tool, it opens in the middle, that blue panel for running tools and viewing data. And this is really where you interact with things in Galaxy. And so for the tool, it'll give you various parameters. It'll explain what you might want to set the parameter to, give you information about the options. And on the right, you have your analysis history. So that's in pink. And this history pane, whenever you upload data, the data will be there and you can view or inspect the data. That'll open in the blue panel. And every time you've done something, it keeps a record of it. So Candace was talking about how we want to work on having more reproducible workflows, more reproducible work that we can share with others. Galaxy allows for that because it keeps track of everything you've done, and it allows for you to export that history in a clean way and share that with other people. So what are the benefits of Galaxy? I've already discussed 4 of them pretty in depth, that you can use a web-based UI, that there's lots of public data accessible, or you can upload your own data, and that they have lots of informatic tools available. It's a widely used platform. It's been used in over 11,000 publications, and it's fairly easy to collaborate with others on your analysis using Galaxy. I've also slightly talked about this, but I'm highlighting it and bringing it back up because it's one of my favorites, that it's easy to track your steps for reproducibility's sake. And so it's easy to share your methods with others given all of this. And let's say that someone is telling you about some analysis they want to do, or you need to test out a tool and you don't really want to have to bother with installing that tool locally, and you just want to see if that tool will work with your data. This is another big benefit of Galaxy, that you can test out that tool. You can see if it will do what you need with your data before you even try to install it locally. So Galaxy has a lot of benefits for you to try out things, for you to have reproducible research, for you to have access to tools that are going to work because they're supported. And in case they don't work, there's lots of help available. There's a support forum, and they also provide tutorials and trainings for common workflows or common analyses you might want to do. So when you're talking about Galaxy, sort of how we were talking about cloud computing, having servers that are distributed all around, well, Galaxy takes that a step further. It has 3 clusters of the clusters. So it has Galaxy Main, which is the main server that you're probably going to interact with. Galaxy Europe, where there's a lot of servers in Europe, and most people in Europe are going to use those, and Galaxy Australia. So this is an international effort. And just because you're in the US doesn't mean you can't use Galaxy Europe. I had to once because I wanted a mass spec tool that was only available on Galaxy Europe and it wasn't available on Galaxy Main. But this is an international effort where scientific communities from around the globe are working together on this. One of the other servers is Cancer Galaxy, which is the server that you will be using today with Hannah as you walk through exploring your microbiome data. And Cancer Galaxy is an example of specialized servers that Galaxy does apart from Galaxy Main, where it's bringing together tools that are useful for cancer analysis or specific to cancer analysis. You don't have to be working with cancer data to use Cancer Galaxy, but it is a specialized server. And here's an example of other specialized public servers where there's one for marine science or frog. Like there are lots and lots of examples of specialized servers, and it's probably good that I don't know exactly what all these work on. Otherwise, I would have way too many PhDs. So is Galaxy free? The answer is mostly yes. It gives you 250 gigabytes of total disk quota, and it's going to be pretty difficult to exhaust that. But if you need to exhaust this or exceed this, what do? You talk to Galaxy Help, or you consider alternative platforms like Anvil, which also brings in protections for data. So as was mentioned earlier by, I want to say, definitely Candace, possibly Carrie too, because we love talking about this. If you have PHI or PII and you need it to be like protected, Anvil's a good thing to look at. And so if you need an alternative to Galaxy, Anvil has similar help forums and guidance on how to get started there. Here is an example of a team that used Galaxy and they were able to provide their workflows to the public through Galaxy so that everyone could see what they did. And so this was a very reproducible example of them using Galaxy in their research. So let's actually look at Galaxy. Please visit cancer.usegalaxy.org. One thing to note is that I thought until about half an hour ago that you needed to register and log in to use Galaxy. It turns out you don't need to. They will let you upload data and run tools and keep a history of it if you're not logged in and registered. However, if you want to use Galaxy frequently, if you want to have multiple histories and you want them to all be available to you, if you want to utilize other people's workflows instead of picking your tool and taking the output of that and putting that into another tool, then you're going to want to log in and register because it gives you so many more possibilities on what you can do if you are logged in and registered with an account. Is anyone else having trouble getting into cancer.usegalaxy.org? Yes, okay. Hey. All right, so don't worry if it takes you a little bit to get there. It seems we're all having that problem. But what you'll see on this top banner is login or register. If this is something that you think you're going to be using a lot, log in or register. Don't worry about it right now. The stuff Hannah's going to be doing with you do not need it right now. Also of note, though, this is Cancer, that specialized one I was telling you about. Main Galaxy, I also learned half an hour ago, you need a different login. I thought my Main Galaxy login would work on a Cancer. It doesn't. So I used the same username and password. But oh, Carrie's telling me not to do that. All right, I'll go fix that. Don't do that. Yeah, just make sure you log in or register differently. And here what you'll see is on the left, we have that tools pane I told you about with the searching available and an upload data button in the tools pane. It's okay. That's fine. It's a little bit different from Main. Main has like a secondary panel like over here where that's where upload is, but that's all right. This is really close to it. And then this is your main information. And if I were to select a tool, it's searching. It brings up the information about that tool in the middle. And if I were to upload anything or actually run a tool, it would keep track of that in my history there. So let's say that you need help. That's where this help button is. There are interactive tours available. I would click this button, but I fear it's going to take too long to load. There are interactive tours available for you if you want a tour to better understand the user interface. And there are also trainings available So various training materials available. This learning pathways is really cool because it'll give you like all of these trainings strung together in what order they suggest if there's a specific topic you're interested in. So with that, I will hand it over to Hannah.
Hello, everyone. My name is Johanna Hagen, and I'm going to be, introducing the metadata and our study and getting you started with your first few commands on Galaxy. Yeah. Like a little, like there? Like closer to me? Oh, okay. Maybe I need to adjust. All right, let me just find my... All right, sweet. Yeah, so like I said, the tutorial data, we're going to be working with a study regarding the hematopoietic stem cell transplantation and fecal microbiota transplant. Yeah, so we'll talk about the study, then we're going to go back over to Galaxy and we're going to import the metadata, examine that, and then do the same with our feature table. All right. Let me take a slideshow. All right, so this is our first article that we are going to be using, and this is a compilation of longitudinal microbiota data regarding stem cell transplant patients. Longitudinal meaning that the samples were taken over time with these patients. And yes, so this study was huge. It's a huge compilation. Over 12,000 samples and over 550 million 16 rRNA sequences. And we will just be using a tiny bit of that, but it is a very cool compilation and a very great resource for the medical community. All right, so we have our bone marrow stem cell here doing its wonderful, beautiful work. So what is an allogenic hematopoietic stem cell transplant, or allo-HCT? So it is going to be donor stem cells that are infused to replace the bone marrow, where they will ideally begin to create new blood cells for the cancer patient or the recipient. To prepare for this procedure, the patient undergoes heavy chemotherapy and a very wide range and long range of antibiotics before they undergo the transplant. And this is done so that they have kind of a suppressed immune system so that their body will accept the stem cells. But it does obviously disrupt their microbiome. And this is called decreased richness, and that means that there are less individual microbes than there were before they underwent this treatment. And patients who lose microbiome richness have a higher rate of transplant-related death and other complications. So it's a pretty serious thing to decrease your richness during this treatment. Okay, so this is from that first study. And so two things that I wanna, before we go over our super cool graphs, are understanding the difference between infusion and engraftment. So infusion is the actual medical procedure where the stem cells are given to the recipient. The donor stem cells are given to the recipient. That is the infusion point. And that is going to be our, that dotted line, day 0 here, that goes all the way through all 3 graphs. And then engraftment is going to be the response to the infusion, and that is going to be the point at which the neutrophils begin creating, they become infused cells and are replacing the cells that the individual was missing before. So yeah, let's go over these graphs. So we have A here, and this is going to be the doses relative to the stem cell infusion days. And so we have, we can see that they start their medications and they kind of peak right before day 0. And then they actually continue taking antibiotics and other, antibiotics through 100 days, which if you've ever taken antibiotics, it's normally like 2 weeks if you have a normal infection. So if you can imagine 100 days, that's a long time of taking antibiotics. And so, yeah, this next one is diversity relative to the day of stem cell infusion. So these blue dots here are before engraftment, and then these red dots are when those neutrophils are becoming infused cells. And you can see that we have a nice clustering up here, which is high diversity, and then they really do see a drop. And then people start to come back up, but there's definitely not as solid of a clustering. It's hard to come back. So, and then down here, again, we have diversity in the blue, and then the engraftment in the red. So our diversity after that peak of antibiotics starts to trend down, and then we see people start to come back up, but they don't come all the way back to their previous level. So we can see here that this is a problem, right? We're not recovering the way that we might need to, and this has medical implications as well. So the answer to this is this next study, this question of can we use the fecal microbiota, can we use an auto FMT to treat as a single intervention this problem? All right, so what is an autologous fecal microbiota transplant? So a regular FMT is the transplant of the gut microbiota of from a donor to a recipient to establish that donor microbiome in the recipient's gut. But an auto FMT means that the donor and the recipient are the same person. And so you might think, well, what's the point of that? But the point of this is that with these patients, we want to reduce the risk that they will come in contact with any sort of pathogen that could cause them harm, because they are severely immunocompromised at this point, right? Which is why, yeah, we need this. So, yeah. Okay, so that brings us to our study. We had 25 patients who received who were randomized into control and treatment groups, and they received their FMT 8 to 27 days after their stem cell transplant. And then they were able to see that the study, it was successful. They were able to, we can look at that. So here we can see how this works. This is illustrating one representative patient. But again, this top graph, we have the medications that the patient was prescribed. And first, I want to show these dotted lines. So day 0 here, that goes all the way through, is going to be the day of their stem cell transplant. And then this is the day that this person was able to engraft those stem cells. You can see here, they see that they're starting to trend up. And then this dark line here is going to be when they receive their auto FMT. So this first graph up here, we can see what medications they were actually prescribed and how long they took each of them. And then B is the neutrophil count, which is a white blood cell. It's like a representative white blood cell for a stem cell infusion. And so we can see that they're here, and then they drop off after this, and then they start to come back up and trend a little higher. This is the most interesting one, in my opinion. So you can see here, this is going to be their donor sample. And then they start their antibiotics, they continue through. And so these are microbial taxa composition plots. So you can see on the key here, these colors actually indicate which microbes are present. So in a diverse microbiome, you want to see many different colors, right? So as they proceed through their antibiotic regimen, you can see that it's turning less and less rainbow colored, more and more green, and then just green and purple. And the very cool thing that you see on the day of the FMT is they get that diversity back. And even though they are continuing through antibiotics up here, on day 91, they still have a pretty good diversity continuing through. And so that really shows you in color what that could look like. And then so down here, we have two graphs that show us more about the diversity and how that trends through. You can see it goes down and then comes back up after FMT and kind of stabilizes up above. And I think that's pretty cool to see. All right, so are we ready to do some commands? Okay, let's do it. I am going to come out of here somehow. There. All right, so if you could go back to cancer.galaxy.org, I believe it is. Use Galaxy, thank you, yeah. And actually, yeah, we need to open our tutorial. Oh, am I frozen here? Oh, I see. Sorry, the Zoom menu is in the way always. There we go. Yes. Oh. Right here. Oh, okay, good. Okay. And then do we have our tutorial book here? Okay. Thank you. All right, so I'm gonna also open our tutorial, if it will let me. And if you would like to also as well follow along with me, open your tutorial, and then we can go together through all of this. But let me make sure I'm here. Yeah. Yes. All right, so here I'm at the very top of the slides. If you go back to the PowerPoint. And then at the very bottom, it says tutorial with a semicolon, and then you're just gonna click on here and it should give you a link to follow. If you need more time, just raise your hand and I can wait. All right. And then over here, we are in our tutorial now, and then I'm gonna also have my Galaxy window open as well. I'm gonna scoot it over. All right, and that's cancer.usegalaxy.org. Does everyone have those two tabs open? Just raise your hand if you need more time. Oh, you're good. Okay, cool. All right, so first things first, we're going to go to the tutorial itself. And over here, we have our options, our little table of contents, and you're going to click exploring the data with Galaxy. And then scroll down a little bit, and this is going to give us our instructions for how to get our data into Galaxy to begin with. And so if you want to play around with this later on, you can always come back to this tutorial and read through it on your own and get to this on your own. But we're going to go over to Galaxy now, and we're going to click upload data. All right. And then paste/fetch data. And then let's go back and make sure we name it the right thing. So we're going to want to name it sample-metadata.tsv. And then we're going to go back and copy that link. Great. And then in this big box here, you're going to paste that link. And just as a little check, I always like to make sure I didn't accidentally miss any characters at the beginning or the end because it won't work. So just double check that you have everything there. And then we're going to go ahead and press start. And then hopefully you saw this pop over on the side here. And once this turns green, you can go ahead and just press close. And now it's going to be yellow for a little while, and then hopefully it will turn green. And that will mean, oh, cool. So that means that our data has successfully loaded in. Thank you, Greg. Does anyone still have yellow for their metadata? Wow, that's amazing. That's really great. Okay. Everyone? Okay, cool. All right. So now let's go back to our tutorial and make sure we're following along here. Yeah, so we got our stuff and now we're going to run our first QIIME command. So that's going to be QIIME2 metadata tabulate. So to use that command in Galaxy, we're going to go back to Galaxy. And as you can see, I already have a bunch of QIIME tools open over here. And lucky me, my metadata tabulate is right at the top here. What you can do if you don't see it here, let me make it more difficult for myself here. All right, so this is probably what yours looks like. You're going to go to the bottom here and it's going to say QIIME2 tool suite. You're going to click on that and that will open up the whole menu of the QIIME2 tools that are available. And then you can also search in the tools here. So I know that I want yeah, what is it? Metadata tabulate. Yeah. Yes. Oh, yeah. Great. Okay. All right. Metadata tabulate. There it is. All right. So I'm going to click on my tool and then hopefully we all see this here. If not, so this is obviously our file that we just loaded in. So this name should match this green name over here. And you're going to hopefully have it here. If not, you can select it from this drop-down menu. If you don't see it here, you can click here and it should show up as well. But it preloads those things for you most of the time. So hopefully we've all got it there. Raise your hand if you don't see your sample metadata. Great. Okay. Go ahead and press execute. And so that first gray moment is your job essentially waiting in the queue. And then once you see this ... going around, it's actually gonna start running soon. So, or it is running. So we just have to wait for that to go and then we will be able to look through. Yeah. Yep. That's our next step. I know the anticipation is killing me too. Yeah. Wow. That would be, I think that's a great idea. I don't know if they have that, but I think that would help a lot while you're waiting, to be able to watch your job kind of progress. Yeah, totally. Right? Like, hey man, you've been on here all day. It's my turn. So metadata tabulate is going to actually show us what our metadata actually looks like in QIIME. So it's loading the metadata file that you have, that you made or received, and what it's going to look like to QIIME. And it's going to give you lots of nice information about it. So you can have your metadata in a TSV or a TXT. Those are the two most common formats that you'll have it in. It's normally like an Excel file, just, yeah. Yeah, I think CSV is totally fine as well. It's just, how it's separated essentially in the file. Is anyone's job not yellow? Is everyone's job yellow? Cool. Nice, you beat me. All right, so we should start popping up soon. Yeah. Yeah, yes, absolutely. Yeah.
So while we're waiting for this, I'm just going to show you one other section of the tutorial book that we put together. And this is what would be great for folks to do at home tonight. And so if you see this next section here that says working with your QIIME2 container, this contains the instructions for getting the Docker container or Podman container running on your computer. And so it would be great if you could, like when you get back to your office today or when you get home, just run through the steps in this section. And I would go through this interact with Jupyter Lab and QIIME2. And basically, this is just going to have you run a few test commands after it's running. If you go home, you try this, you get stuck, don't stress about it. We can help you with it tomorrow. If a few people are downloading this while we're here tomorrow, it's probably going to work just fine. But like I said earlier, if everybody tries at the same time, we're going to have some problems. Just again, this is that working with your QIIME2 container section. And we're green.
All right. So I am going to go ahead and continue while the rest of yours are popping up. Yes. Oh, okay. She's getting up. All right. So you're going to go once it turns green, and you can let's change this name first, because this is quite a mouthful. I'm going to go over here and just change this to, well, I think it tells us what to call it. No, I think you're good. Yeah. Yep. Yeah, so down here it tells us what name we should call it. Oops. Okay. Metadata dash sum dash one. All right, and an important part of this is this QZV. You will see in QIIME QZV and QZA. The V indicates that it's a visualization, and then the A will indicate that it's an artifact. And so, yeah, that'll be important for later, but I'm just gonna save this name so that it's easier to remember, and it helps you stay organized if you have good names on your artifacts. All right, and then I'm going to click here once again, and it's going to open up this menu here, and then down here it says view at QIIME2 view, and we're going to click on that. All right, so here we have our metadata, and yours will look the exact same as mine. So I'm just going to give you a little tour of our metadata for this workshop. So as you can see, we have kind of an Excel file looking artifact here, and you can scroll across and see all the columns that you have. And if this is your metadata, this is a good time to make sure that all of your columns are here and that things look correct to you, because you're going to be working with this data for the rest of your workflow. Yeah, it's very important that things are looking correct to you. Yes, and then something else to note in QIIME is these tags at the top. You can see it says numeric and categorical. So these are just the way that QIIME decides what kind of column you have. So QIIME will infer what type you have if you do not put them directly in there. So the way it does that is if it's just numerical values inside of the column, it will say, oh, this is a numeric column, and it will label it as a numeric column for you automatically. And then if it sees alphanumeric or anything else besides numeric, it will label it as categorical. Luckily, if you want to override this, you don't want QIIME to do it, you can do this yourself, and you can set these before you load it in or at any point. All right, so the first column that I want to kind of underline here is our patient ID column. So here is our patient ID. This is our unique identifier for this patient, and it allows us to track this patient through time. You will see that it's kind of odd, right? We have, if this is the unique identifier, we have how many of these people in here? Well, these are actually samples from this patient. And this is an opportunistic sampling study. So whenever the patient came in was when they sampled. These time points are not standardized. So whenever the patient was at the hospital or wherever they received treatment, they would give a sample if they could, and those became our samples. And then let's see. Yeah, you can see which group they were in here for the auto FMT group. All right, the next very important column is the donor sample ID. And it looks identical or almost identical, but you can see that it has the A at the end here. This is the actual sample that is going to be donated to the recipient. So this was the sample that the patient gave that is going to be donated back to them once they have gone through their stem cell transplant and have stem cell engraftment occurring. So out of all those samples that we see, this is the one. And this is very important for later in our analysis later this week when you are going to use Q2FMT, Chloe Herman's plugin. This is a very important identifier for that plugin. All right, and the last column that I want to highlight is the time points column. And so as you can see, we have some intermittent numbers here, and these are the, so 0 through 2 are the time points before the auto FMT is administered. Day 3 is the day of the auto FMT treatment. And then any numbers after that are going to be after the FMT. And you will be using this column later in analysis as well. All right. Does anyone have any questions about the metadata right now? So we're going to go back and run our next command. All right. Let's go back to Galaxy and back to our tutorial. Okay, I'm just going to save my name. Raise your hand if you're still seeing yellow for your visualization. Okay. You should be next because everyone else. All right. You can still follow along with me. Because you're just loading the visualization, so you can join with us. Even if your visualization is still loading. So go ahead and click upload data. And then we're gonna, again, click paste and fetch data because now we're gonna load in our feature table. So going back here, we're gonna scroll down a little bit to access and summarize the feature table. And okay, we're gonna name it feature-table.qza. All right, and let's go grab that URL. And then paste it into the large box at the bottom. And you don't really need to worry about any of these other pieces, just as an FYI. And press start, and then that should be loading in. All right, and while that is loading, I'm gonna look up my next command. And that's gonna be feature table and summarize. Here it is. Feature table summarize is gonna be our next command. All right, and hopefully your feature table is loaded, and hopefully it's also seen here. Oh, something else I'd like to mention is that these numbers here, I think they probably are matching up with yours, but this is kind of just your Galaxy history of artifacts number. And so this number is not actually attached to your files. It's just inside of Galaxy. You'll have those numbers on there. Okay, so don't press execute yet because you need to add in one more thing. We need our metadata. It's very easy to forget your metadata, and if you do, you won't get anything interesting really to see. So don't forget your metadata. All right, so my metadata is already preloaded on here. And then once I'm ready, I'll just press execute. Yeah. Yes, it will. Yeah. That'd be great.
Okay, so again, this is probably going to take a couple minutes. I just want to show you a couple of other things here. And so a lot of times, like if you're working with this on your computer, you're not going to have these links like we're providing. You're going to have maybe files on your computer. And so basically like what you would do in that case is just choose local files. We're just sort of bypassing that, giving you links to where you can download these files from, just so we don't have to distribute all of these to everybody, but you would just go with that option instead. One of the other things that I wanted to show you here, and this is kind of a neat feature of QIIME2, but like you'll see that like if I were to come in here, I just clicked on a random one of these. I clicked on QIIME2 Taxa Collapse. What Galaxy and QIIME2, when they're integrated, what they do here is they know which specific artifacts you have that are valid inputs for different commands. And so like what this one does, so this is a command called taxa collapse. And so what this does is like if you have a feature table and then you have taxonomic annotations associated with all those features, what it'll do is it'll take like an amplicon sequence variant table and it'll collapse it to be like a genus table or a species table. And so it needs two different things to do that. It needs the feature table and then it needs this taxonomic annotation. And so here, like you can see that this has populated in our feature table. And it does this because there's under the hood in QIIME2, we have some annotation of the types of different artifacts. And so under the hood, without you having to be involved, this tool knows that it takes a feature table as input and you happen to have one of these feature tables in your history. If you had 10 of them in there, it would give you all 10 of those feature tables and you would just be selecting which one you wanted to use there. In this case, we haven't done any taxonomic annotation and we haven't loaded any relevant taxonomic annotation artifacts. And so this is telling me there's no data set available for this. And so basically what that means is like we can't yet run this command because we're missing some of the information that's needed for it. Any questions about that? The other thing that you'll see in here is there is this difference between required inputs. And so like another thing that's required here is an integer that specifies level. And so that usually would imply, we haven't looked at all the help text for this, but like that would imply the taxonomic level that you would want to collapse at. And so that's going to be a numeric indicator of like phylum, class, order, family, genus, species. And so we have this covered in some other tutorial content on docs.qiime2.org, but I just wanted to mostly show this to give you a view of how this looks through Galaxy. The last thing I'll mention while this is going is we've just introduced these two different file types that if you haven't used QIIME2 before, you're probably not familiar with. And that's this .qza file and the .qzb file. You can find lots of info on those in our documentation, but what a QZA file is, it stands for QIIME zipped artifact. And so in other words, what that is it's a zip file that has some specific internal structure that QIIME2 knows about and QIIME2 expects. And so you can open one of these files with any unzip utility. So you can like call unzip on it. You can use 7-zip or WinZip, whatever you would normally use to open a zip file. You can just feed this in and it'll open it up. And if you were to open it up, you would find a data directory in there. And in that data directory, you would find your data in the typical format that would be associated with whatever artifact type that is. And so, for example, if this was demultiplex sequences in a QIIME2 artifact, that data directory would contain a bunch of fastq.gz files in it. If this was a phylogenetic tree in an artifact, it might be a NUIC file that you would find in that data directory. So QIIME's not really doing anything magical in terms of like what these files are. It's just that by putting them in these data artifacts, we can associate other metadata with them that is relevant to the QIIME2 system. And so, for example, this is where we store the data provenance. And so any information that we have on how that artifact is generated would be in this unzipped artifact in a directory called provenance. And we did this, like we made the specific choice on how to represent this data because we wanted to be able to put other metadata in there. We wanted to make it so that we could change the underlying formats without the user having to be aware. And so, for example, if we were to come up with a new file format that was able to compress FASTQ files to much smaller sizes, we could swap that out under the hood and you as the user wouldn't have to know about that. You could still convert between different formats and export things in whatever format you want, but under the hood, we could make some changes that make the system work more efficiently. And then the last thing that we wanted to do here was not create some new file format. And so we, like, our minimal approach there was to describe the structure of the zip file, but in a way that's very human accessible. So like I said, you can unzip it, you can poke around in there, and you can find the data that you're looking for. And the zip, like, it by nature of it being a zip, we essentially like wanted this to be what we would call future proof. And so let's say it's 10 years down the road, and I hope this isn't the case, but let's just say, nobody's using QIIME2 anymore. But you come across one of these artifacts because you went and you downloaded some supplementary material from a paper. Even if you can't use QIIME2 to open up that QZA, you could just use an unzip utility like 7-zip or WinZip or something like that. And then you can access that data, even if you can't access QIIME2. And if some future we can't open zip files anymore, then we probably have bigger problems. And so we're not going to solve that one. Okay, and I think we are green. And so I'm going to turn it back to Hannah.
All right, I think I saw a few people in the green as well. So let's go ahead and explore our data. And if yours is still loading, you can just watch along. And then once yours pops up, you can join us. So once again, we're going to go to view at QIIME2 view. All right, I'm going to zoom in a little bit. Okay, so first we have several tabs, or 3 tabs here. We have our overview, our interactive sample detail, and our feature detail. Our overview is really going to give us a bird's eye view of our feature table. So first we have our table summary, and this is going to show us how many samples we have. So if you remember at the beginning, we had 12,000 total samples in that compiled data set. We're working with 556 of them. And then our total number of features, which is 4,256. So this is going to be the ASVs that are represented in these samples. And then the total frequency of these features is 28 million, it looks like. Very close to 28 million. All right, so frequency per sample table here. So you can see how our samples are represented. And then we have our graph over here, number of samples over frequency per sample. So the minimum is 268, and then the maximum is 700,000. But you can see that most samples kind of land around here. And then we'll see that more later in the interactive sample detail. And then down here, frequency per feature. So this has to do with the ASVs. The minimum ASV you'll have is 1, and then the max is like 2.5 million, but the average is about 7,000. All right, and then I'm going to skip over to feature detail because interactive sample detail is really the fun one. So let's look at feature detail really quick. You'll see these very easy to read alphanumeric chains here. And so these are going to be the ASVs themselves. And this is just an indicator of who they are. So you can use this later on. And then you have the frequency and number of samples that this ASV is observed in. So if you wanted to look up a specific ASV, you could do that here. And as you can see, the scroll bar is quite long. So we got a lot of ASVs to look at. Okay, let's go back to interactive sample detail. All right, so this is our number of samples over our day relative to FMT. On this axis, you're going to see whatever you have decided over here from your metadata category. So typically in your analysis, you will have 1 or 2 columns that are really important to you. And so you'll probably want to sort by those. And so let's look at time points because that was one of ours. So we can see that we have a lot of null here, which might be concerning. But if you go back and look at your metadata, you'll see that, wait, they're not all filled in. right? So that's going to be our blanks here. But we can see here that we do have our numbers that we need. And then over here, you're going to see the sampling depth, and you can go ahead and play around with how moving this slide bar here affects who is allowed into your study, essentially. And another thing you can do is scroll down and see how this is affecting as well. So if I just scroll down a little bit, these red will essentially be dropped because they will be excluded with this sampling depth. And we're going to go more into sampling depth tomorrow, I think. So how to choose the sampling depth that is best for this study and any future study you will do in QIIME. And yeah, that is, I think, all that I have for you today as far as your first two commands, but you are welcome to play around with Galaxy. And oh, and then the papers that I mentioned at the beginning, I believe are also linked in here. So if you wanted to read up on those, yeah, here they are. So you can read up on those. And oh, also you can read up on Q2FMT as well if you wanted to really get prepared. But yeah. Any questions about your first two commands in QIIME? Yeah. Yes. There. All right. Yeah. Yes, please.
Okay, thank you so much, Hannah. Okay, so the last thing I wanted to just mention today, so like this is, we gave brief introduction to Galaxy and had you run through a first couple of commands there. And I think we, all experience kind of the issue that I mentioned earlier today, which is, we're essentially using a shared compute resource for this. Um, and this is, particularly slow because what we're doing right now is we're running these really quick steps. So like these things that really should have like a very snappy turnaround. But we're spinning up a lot of computational infrastructure to do that. And so there's some like fixed costs here. And so, for example, like getting the tool loaded on this remote computer that we're using, and then sending information back to the server, those are going to take about the same amount of time, regardless of how long the actual computation takes. And so when we run something like this feature data or like feature table summarize or metadata tabulate, those are really quick things. Like if we did those on the command line with this data, it would probably take 1 or 2 seconds for that job to complete. But we're, like I said, waiting in a queue. We're waiting for some machinery to be ready. If we were to run a job on here that normally would take 12 hours or 16 hours on our computer, we wouldn't really notice that difference probably when we're running it on Galaxy, and we may have the potential to make more use of the high performance computing resources, make more use of the many processors that are available, like Carrie was talking about this morning. And so it's like when we run these little steps, we notice how long it's taking, the overhead of using this. When we run bigger jobs, that overhead becomes negligible. And, it's like you would hit it, and then maybe you're going to go out for coffee and come back in an hour and check on whether it's done, or you're going to start a job over the weekend with the hope of coming in on Monday morning and getting your results. And one feature that we didn't look at, but is pretty neat with Galaxy, is you can actually have it. I think you need to be logged in for this, but you can have it send you an email when it's done. And so that way you can just be alerted, get that information. Okay, so yeah, from there, I just wanted to again mention, this working with your QIIME2 container. What we're going to do tomorrow is we're going to jump in with that. We're going to talk a little bit about, we're going to learn a little bit about what these containers are at a very high level, how they work. Um, and then we're going to do these same two steps in a container tomorrow on your computer. And so that's just going to kind of prime us for diving in and doing some of the diversity analyses and then doing some of the more specific fecal microbiota transplant analyses. And then, like, what I'm hoping you'll see tomorrow is that there's this one-to-one correspondence between how you run something in QIIME through Galaxy and how you run it through the command line. If we go back to that tutorial that we were just looking at, one thing that we didn't play with it at all so far is this little drop down at the top of the page. And so if we click on that, what you're going to see is there's these 4 different options that you have. One of them, the default on this page was Q2 Galaxy. But I'm going to go ahead and I'm just going to click this command line and I'm going to scroll down and you can see that now these commands have been translated into how I would run them on the command line. And so this is where we're going to be jumping in tomorrow. And so that step where we were copying the URL over to Galaxy to download the file, we're going to use this command called wget, which basically says, go download this file from some link on the internet. And then where we navigated through the Galaxy menus and found the QIIME metadata tabulate command, we're going to be running that directly on the command line tomorrow. And so that's going to be QIIME metadata tabulate. And then we're going to do the same thing for the feature table. We're going to call feature table summarize. We're going to give it our feature table QZA, and we're going to give it the sample metadata.tsv. In this workshop, because we have limited time, we're going to start the analyses with these feature table files. Just because a lot of the very computationally expensive steps happen upstream of this. And so that would be doing things like quality control on the sequence data and generating this feature table. We have all of that stuff covered very extensively in our documentation. And so rather than spend time on that in this workshop, we're going to focus on the aspects of the analysis where we get to poke around with some of the interactive visualizations. But we're more than happy to answer questions on those upstream steps, really anything you're wondering with respect to QIIME. Okay, Carrie, do you have any stuff that you want to wrap up with today? Okay, great. So I think do we have time for a few questions before the survey? Okay, great. Yeah. That's a great question. So just to repeat the question, it was, is Galaxy and the container interoperable? So in other words, can I download something from Galaxy and then run it through like in a command line analysis? The answer is yes, they're 100% interoperable. Same goes for the Python API. So, like the way I use this a lot is I will log in on my university's HPC system and I'll run the computationally expensive steps. I'll submit them to the queue on the cluster, let them run, come back, start them up on Friday afternoon, come back on Monday morning. And then I tend to download the files to my computer. And then I'll often use a Jupyter notebook and I'll be working in Python at that point. And so what I do is I use the HPC for the really like very non-interactive steps where it's just kind of chugging away, doing some data analysis. And then when I'm ready to do the more interactive steps, I'll download those, run them on my computer. Those steps tend to be pretty quick. Sometimes I'll run something that'll take 5 minutes or so, but don't need the 12 hours of tying up the computer for it. And yeah, so I'll usually bounce back and forth between doing something in Python and doing something on the command line, even on my own computer. The other nice thing. Oh yeah, yep. Yeah, so that's a good question. So if you want to download this, so like let's say, I'll just show you, yeah, a quick example here. And so if I click this save button here, okay, it just automatically did it. And so if I go ahead and open the folder where it did that, I don't think I have an ability to zoom here. But you can see now I have this .qzv file. One thing that is a little inconvenient is just the way that Galaxy names these files. They end up with these really long names. And so I might locally just call this, I'm just going to call it feature table.qzv. And then I'll use this other interface just real quick to show you. But if I navigate to view.qiime2.org, so this is that tool that we were using for viewing visualizations. What I can do is I can click in there. It's opening this file dialog for me. I go to my downloads folder and I open up that QZ, or I navigate to that QZV, and then it opens it up and you can see, same thing as if we just clicked into that view link. It just went through this step of bringing it down to my computer first. Yeah. Do we know what version Galaxy is using? Okay. So let's see. Okay. Oh, I see. So up here, it's, yeah, showing that this is QIIME2 Galaxy version 2022.11. So this is another point that I want to bring up here, is that like when you're using these servers, you're waiting on other folks than us to update the versions of software that are installed. And so this is actually like a fairly old version. What that means is just some of the newer functionality that we've added is not going to be in here. But if we were to like identify any serious issues with these older versions, we would talk with them and we would make sure like tools were updated on the server. So basically it just means like you're not getting all the bells and whistles, latest and greatest stuff that we've been working on. Now I want to show one other way of answering that question of what version. And so if I come into QIIME2 view, we talked a lot about reproducibility and data provenance today. The provenance of this is not going to be super interesting yet because we haven't done a whole lot. But if I click on that provenance tab in QIIME2 view, then I get our data provenance viewer. And so if so what these represent in here, the circle represents some piece of data, either a QIIME2 artifact or a QIIME2 visualization. And the square around it represents the command that was used to generate it. And so if you were to scroll in here, so let's say that Galaxy server is not available anymore, or it's been a year since you ran it and you're like, oh, wait, what version was I using? All of that information is in here in the data provenance. And so for that, you have to scroll down quite a ways, but you can see it's version 2022.11.1. If we go back to Galaxy and we compare that, you can see that was 2022.11.1. And so under the hood, Galaxy is making a call into QIIME, and then QIIME takes care of all of this provenance recording. And so it makes sure all that information is there. There's also, there's just an enormous amount of detail in here. The vast majority of it you will never need to use, but that one piece of information that you are going to need is, almost certainly going to be in here somewhere. Um, if we were to trace this all the way back up, this is funny actually. I mentioned that these are very like computationally not expensive steps that we're running. The actual time that QIIME took to run that step was 5 seconds. We waited a lot because of like the overhead of interacting with this other system. Again, that's a big deal. If it's a job that takes 5 seconds, it's not a big deal if it's a job that takes 12 hours. And usually, it's the bigger jobs that you're running on a system like this. Um, so like what's happening in Galaxy when we, okay, so if you're like trying to submit new functionality for Galaxy, well, when we so when we run it, like what we do on, oh, okay. Right. So if you want to share this information like for publication, how do you tell others to how to reproduce this? So in Galaxy, you would share your history. And so I believe that is up here. I think because I'm not logged in, I don't have the ability to share history. Oh, was it in there? Oh, export history to file. And so this is going to be Galaxy's perspective on what happened. Um, the way that we typically do this with QIIME2 is we have another tool. We're going to show this to you at the end of the day on Friday. But we have a tool that's built into QIIME that enables you to generate what we call a reproducibility supplement. And so it's basically going to create a zip file for you that has all of the commands that you ran. So like generally you would produce one of these reproducibility supplements on some folder that has your final results in it. And so you did all your analysis and it relies on like these 3 tables and these 2 visualizations, say. Put all of those in a folder, and then you run this command that we'll show you that is QIIME replay uh, reproducibility supplement. I forget exactly. And what it'll do is it's going to create a zip file for you that has all of the commands that it would have taken to generate that as command line calls and as a Python script. And one of the other nice things that it does, I'm not sure if we have any useful information in, okay, yeah, we do. It will also give you a list of the relevant references for the analysis that you ran. And so again, we didn't do a whole lot here. And so this isn't a very big reference list at this point. But when you've run a whole bunch of different things, this sort of facilitates that for you. What's really important there, and the reason why we built this into the framework, is because if you're using QIIME2, a lot of people will think like, okay, I use QIIME2 to do my analysis, so I cite the QIIME2 paper. But like I mentioned earlier, we've got folks all over the world who are contributing plugins, who have built methods that we're relying on. And so when we write those plugins, we integrate information on the relevant citations in there so that if you generate this reproducibility supplement, you get a list to make sure, okay, I cited Chloe's Q2FMT paper in there because I was supposed to cite that one because I used it. That's it's important for you for your work. It's probably most important to our plugin developers who are, want to get credit for their work. And so we try and make that really easy to help motivate people to build for the system. Any other questions? Okay, then I think we are going to wrap up the day with a survey. Okay.
So we have just like a couple quick announcements and survey things that we hope you can help us with.